AIM-listed ValiRx is a life sciences company focused on early-stage cancer therapeutics and women's health. It recently created a subsidiary called Cytolytix, which has signed an IP license agreement with a highly respected King's College London. 60% owns Cytolytix will take a potential breast cancer treatment called CLX-001 through preclinical trials. Also, as many of you know, ValiRx is waiting for TheoremRx to raise funds in order to license VAL201 for oncology purposes. Suzy Dilly, CEO, joins us live to explain all in more detail. Welcome, Suzy.
Thank you, Donald. Good evening. I was worried how I was going to fit the presentation into 20 minutes, but I think you've covered most of it in the 2-minute introduction there, so thank you.
I know, I've hogged your glory, Suzy.
So it saved me a job. It's easy when the introducer does all the work for you.
Okay, I'm sure you're just being kind. Over to you. Let's see what the real company story is.
Thank you. So as Donald has mentioned, tonight we are introducing Cytolytix. So this is our new subsidiary company recently set up with King's College London. But before we go any further, the first thing I have to do is to just highlight the disclaimer to you. So this presentation will be available in PDF form on our website afterwards, as well as hopefully a link to the recording at the end of the presentation evening. But we do just have to highlight the disclaimer. Please do your own research. Don't rely solely on the information given in this presentation in order to make a decision about any investment. But moving that aside and getting on to the more exciting things, hopefully there are a few members of this audience who have seen things from us before, but if you haven't, do feel free to look on our website to see presentations that we've given in the past, because actually we're not going to go over too much of the old stuff today. We're going to focus on the new company, on the new science behind the new company, and just briefly touch on some of the other things that have been going on within the company over the last few months, and in fact the last year. We have been very busy. And you can see from the at a glance slide, which is designed to give a summary of everything we're doing. We did complete the fundraise in the summer. So at the end of June, we did raise 2.5 million. So thank you to everyone who participated in that. We do have 2 programs that are currently undergoing out licensing. So these are programs that have been taken through into clinical testing. That isn't our intention anymore to take programs into clinical testing. We are focused solely on in order to reduce that cash burn, diversify the portfolio, and to make sure that we have plenty of new early stage pipeline projects coming through. Those pipeline projects that we bring through are focused on oncology and women's health. Now, we do find that there's a lot more out there for innovative science in oncology than there is in women's health at the moment. And that's really why we are focusing on both of these 2 areas, because we want to change that. We want to make sure that women's health is brought to the forefront of biotech companies' minds, and that tools are created and used and made available to help push women's health projects forwards. I will be talking about our evaluation projects. I'm not going to be talking so much about our TCRO strategy. That's our translational contract research organization idea. If you want to know more about that, please have a look at some of our presentations that we've given over the last 6 months. But just as a very brief line on that, we believe that there is a gap in translational services, so taking projects from those early preclinical stages out of universities, translating them into industry, and then making them ready for that translation from preclinical to clinical. Now, that's what we're doing with our assets. But we want to make sure that if we find good technologies that enable us to do that, we can also offer those as a service to bring in revenues for us, but also to enable more good science to happen in other companies and not just ValiRx. But the focus for today, as we've already said, is Cytolytix. So Cytolytix is formed, the name is formed exactly for the science. So cyto meaning cell and lytix meaning breaking apart. This is a molecule, a drug candidate that can break apart cancer cells in order to treat patients who are suffering from that cancer. So I will go into a little bit more detail about the science in a little bit, but that's just to give you the headlines. So who are we? We're ValiRx. Again, if you want to know more about all of us, please look on our website and at past presentations. But particularly to highlight here are two brand new members of staff taken on since the AGM. We have Stella Panu coming onto the board as a non-exec director, in replacement for Kevin Alexander, who chose not to stand for re-election at the AGM. Stella has come in with her vast experience of institutional investment and her network of contacts that will help us to be able to build that more sophisticated shareholder base that we are hoping to build. And really excitingly, we have Kathy coming in as Chief Scientific Officer. Now, she is interim Chief Scientific Officer at the moment. I'm hoping that that's not going to stay that way forever, but she brings her experience of really extensive experience of translational science. She's worked in universities, top class universities. She's worked in big pharma, GSK, Takeda, for example, and she's worked in small biotechs. And specifically what she's been doing there is taking early stage innovation and bringing it through just exactly how we do. So this is really a brilliant match, and I've enjoyed the last few months working with her. I intend to enjoy a lot more of that. So where are we at the moment? Well, we have a new pipeline picture to share today. This is the first time anyone's seen this. We have the green line at the top for Cytolytix being our new program that's come in. It's gone through our evaluation stage where we first take projects from universities and we test them for about a year first to make sure they really do what we want them to do. And we found that CLX 001 did do exactly what we wanted and didn't do anything that we didn't want it to do. So we formed that subsidiary, Cytolytix, and that is now looking to move into more formal preclinical development. The other programs that we have on here, the two evaluation agreements, Hokkaido University project and Barcelona University project, those are both still undergoing evaluation. Hokkaido University, that evaluation period runs out in December, so we'll be providing an update before then to see exactly where we are going with that project. And with Barcelona, that update will be provided before the year is up in February next year. The VAL 301 and BC 201 programs are in preclinical development at the moment. So I'm not going to be focusing on those today, but when there is new science coming out of that, we will share it with you. And then finally, just touching on the two legacy assets, the prostate cancer and the lung pancreatic cancer projects, they are looking for, but 401 is actively looking for a partner. It is in an intensive business development phase at the moment. And VAL 201, of course, has the exclusivity agreement under the letter of intent with TheoremRx. So hopefully that should be resolved soon too. Now, just very briefly touching on where we operate in the drug development cycle. This 20 years represents the 20-year patent life. We focus just on that bit at the beginning, that preclinical section. But we always put this slide up just to remind people that it's really important to get every stage right, because every extra month we take now, even back at preclinical, impacts the amount of time that the eventual commercialization partner gets to sell the product. So it makes the product that bit more expensive if we can't get it right at the beginning. And what we are looking to do when we take the early stage academic research and progress it to the point where it's ready to go into clinical trials, we have to get it over this valley of death. We have to make this bridge. And this is just a reminder of all those things that we need to do in order to get it across that valley. So Cytolytix is just at that stage now where we can start filling in some of these gaps. And I'll talk you through in a minute exactly which of those gaps we still have to fill, which actually is most of them, because we are right at the beginning of the process with this one. But now changing the scene a little bit, changing the background colors, and on Cytolytix, the new green slides for the new company, CLX 001. This is a summary of everything here, but I will go through these points in separate slides to get a bit more detail in just a moment. But as I mentioned earlier, the idea of CLX 001 is that it can smash cells to pieces in order to beat the cancer. And most importantly, it can do that without the need for any specific cancer cell markers, any receptors or surface markers. And it does this by recognizing the tumor microenvironment. So it's an ionic interaction that's been optimized specifically to only have this effect on the cell membranes when it's in a cancerous microenvironment. So this is absolutely brilliant science. This has been developed by Martin Olmschneider at King's. We have his research paper. There's a link to it available on our website if you want to read more of the science detail, but I will walk you through a little bit more of it in a moment. So just to think first about what cancers this could be useful for. Now, as it doesn't need a particular cell receptor or surface characteristic other than this tumor microenvironment to act, this could be applied to a lot of different cancer types. Now, we are looking at solid tumor types. It is specific to the solid tumor type that we get the right microenvironment for it. But we've started with this, as Martin Olmschneider recommended, with triple-negative breast cancer. This is very well recognized as one of the more aggressive forms of breast cancer, and it is pretty much the hardest form of breast cancer to treat, specifically because it is triple-negative. It doesn't have estrogen receptors, it doesn't have progesterone receptors, and it doesn't have HER2 receptors. It's where the name comes from. It sounds a bit obvious, but you need something like CLX-001 to come in and be able to smash that cell to pieces, even though there's no cell surface receptor there. So what's really important about this is that although triple-negative breast cancer can be diagnosed as such as soon as a patient is diagnosed with breast cancer, it's actually also something that can happen on relapse. So if a patient is treated by targeting one of these receptor types in the early stages of their disease, they may have a number of years of cancer-free time, but then when the cancer comes back, they can find that it's come back with a different phenotype. And that could mean that it comes back without the receptors because it's become tolerant to those treatments, and those receptors are no longer expressed on the surface. So it is a really important target. And just as importantly, because we can treat without knowing which receptors are there, it opens the door for people to be treated without this type of testing first. So if the testing isn't available to find out what tumor type you've got, you could treat with CLX-001 anyway, because it doesn't rely on those markers. So it is a massive unmet medical need. And it is really exciting to have a potential solution to this problem. But looking a little bit more at the drug candidate itself. So this is a peptide. Peptides are something that I have some familiarity with. So it really is absolutely within my sweet spot for what I like to develop. And I know that there will be a couple of members of the audience chuckling at that because I do keep coming back to peptides. But this is a synthetic peptide, so it is a chemical entity, not a biologic. It is 15 amino acids long, and they are all naturally occurring amino acids. Now, what this means is that it is quite straightforward to manufacture, and there shouldn't be any massive surprises in it because it's the sort of compound that's recognized by the body as being something that should be there, not something that needs to be immediately thrown out. So King's did look at how to optimize this, how to get the best anti-cancer activity out of it. And they had 3 positions in particular that they varied in order to screen 36 peptides, so a decent number to get the best candidate. And that was EEK. So that was the 3 positions where 2 E's and a K. And there are some backup compounds if there was a problem with this, but there is no suggestion at the moment that we should need those. But importantly, this is a slide to demonstrate to us how it works. Now, there is a presentation on our website of Martin Olmschneider actually presenting this science that he carried out. So if you want to see more of the technical science details, I really recommend you watch that, because there's nothing better than seeing science presented by the people who actually did it in the first place. But to get my take on this, you can see the peptide molecule coming in at the top on column A. And this is representing really that the peptide is presented to the cancer cell. There's a few of these peptides floating around. And as they have that ionic recognition on the cancer cell membrane, you can see a few of these peptides gathering together as more of them get collected in this particular microenvironment. And when that happens, when you get a few of them together, you start seeing them perforate the cell membrane. They come together in order to perforate it. And when that perforation has gone all the way through the membrane, you get the aqueous pores forming. And that's what causes the cell to be smashed to pieces. That's what makes the contents of the cell come out. And that's really how it works. So hopefully that explains with a reasonable selection of pictures so that we can see exactly how it's going to work. And this does also offer opportunities for combination as well, because you've got cancer cell contents floating around for the body to recognize and pick up if it's encouraged to do so. Or you've also got huge great holes in the side of cancer cells that other drugs can get in through. So there are a lot of opportunities around this. But looking now at what we've got to do and what we've already got here at the moment. Now, there are already in vivo assays, so mouse models, that show that tumours can be completely eradicated after treatment with CLX-001. And this is where it has been published in Martin Arnschneider's paper. So this is absolutely great data, but we do need to build on that. We need to look at different tumour types. We need to look at slightly different formulations as well. But the fact that even after a first look from the academic, these mice remain tumour-free for 200 days. And if you look at the length of time a mouse usually lives for, that is absolutely remarkable that they got that data off such a one-shot experiment. But also the in vitro assays, so this is cell line-based assays, where both ourselves in our evaluation agreement and work that was done previously by King's shows both ovarian and breast cancer cells responding really well. And this isn't just in traditional cancer cell lines that might have been in a freezer for months. This is in patient-derived cell lines as well. So this is in PDX models. So this is really great. We have done a little bit of extra toxicology as well, a little bit of immunogenicity testing just to make sure, because it's a peptide and it's in an interesting nano formulation, we had to make sure that there wasn't anything unusual happening with that. But we do have a lot more work to do. Firstly, we do need to optimize the nano formulation. At the moment, it works absolutely fine on a lab bench and on a scale for a university, but we need to get that into an industrial process. And that's really an important part of what ValiRx wants to do with early stage programs, is to take them from academic work and industrialize them. So that is exactly what we're doing here. And then once we have the new formulation, we'll be able to look at optimizing the dose schedules and looking at a whole range of different tumors in these models and going through the formal preclinical assessment so that we can bring that through and get that ready to go into clinical trials with a partner who will take it into clinical development for us. So this is really exciting, but it is a very, very early stage project for us. And it's one of the reasons why we have been looking at the translational contract research organization concept, because at the stages it's at the moment, it would be really great to put this into our own lab and to actually have that complete control and complete freedom of ideas to experiment in our own lab with that. So we are still looking for ideas on how to do that, whether acquiring a fully functioning company that already has revenues, or even setting up a small lab of our own in order to carry out this type of work, with potential to also offer services out as appropriate. But I'm going to stop talking now because I can see that Donald has questions to ask me. But if there isn't anything that I've covered, or if there's anything that I haven't covered tonight, or you don't get your questions in, please feel free to email me or any of the team using the email addresses shown here.
Okay, fantastic, Suzy. That was absolutely brilliant. What a brilliant summary. And 1 minute early, 19 minutes. You're a complete star. I'm gonna give you an easy, unchallenging start. James M says, not really a question for ValiRx. He just wants to give you feedback. That as shareholders, we're all really excited about Cytolytix, CLX-001. Congratulations on the deal with King's, the new direction. He likes the new direction, and the newly appointed team members. Looking forward to future success.
That's absolutely brilliant. Thank you very much, James.
I do this a lot, and most people don't actually. They're not actually so kind. So I take it basically.
No, I think this shareholder excitement with Cytolytix is brilliant, because it's something that was really exciting for us. So to be able to get that excitement over to the shareholders and for them to share it is absolutely fantastic, because it is a really big thing for us. It is what we set out to do when we said, oh, we're going to refocus on preclinical. This is exactly what we set out to do. So it's really brilliant to actually have it.
Let's get to a question. Paul Newham asks, he'd like to know how long it's going to take CLX 001 to become ready for phase 1 trial.
Well, so we would expect it to take somewhere between 2 and 3 years for that to happen, but it is very variable. And I will completely hold my hands up here and say, I'm going to say 2 to 3 years, but as soon as we start doing those first experiments, there is always scope for things to be either speeded up or slowed down on the basis of the results we get, because we want to make sure the right trial is run. We want to make sure the right data is going into that trial for the right patients to be getting into it. And at the moment, the first step of what we're doing is actually to do that stage of planning. So to start planning that first clinical trial, even though ValiRx isn't going to run it and ValiRx isn't going to sponsor it, we have to work out what that first trial will look like so that we can do the right preclinical experiments to support it. And at the moment, I don't even know if that would be a healthy volunteer trial or whether that would be going directly into patients, because we have to build the information around the safety and the tolerability preclinically in order to inform that decision and then backfill all of the data that we need.
And it's a very, very broad brush question at this point. And you've made it very clear that you've got a long way to go to get through the valley of death, and there's all sorts of different variables. But how much approximately might this approach take? What's the capex?
So we would typically, you would expect a preclinical program to cost anywhere from £500,000 to £2 million to complete. So we will be applying for grants, for example, from Innovate UK or from any other bodies that could appropriately support this sort of early stage innovation. And there will be a lot of variables. The type of nanoformulation, for example, that we go with could have quite a lot of bearing on how much toxicology we have to do. If we go into healthy volunteers, we might have to do some extra reproductive toxicology, for example, in order to make sure that we can bring relevant women into those early stage clinical trials. So a lot of it will depend on the end goal.
And Nicholas Slater asks, if someone were to offer you £50 million for a CLX 001 as it stands, would you accept, or have you got much bigger plans for it?
Well, is it just a one-off 15 million, or is that 15 million plus some royalties further down the line? So I think if they were to offer it, I would certainly negotiate with that person. If they could immediately see that level of value, that would be a door to open the negotiation. I wouldn't say I wouldn't be biting their hands off if there wasn't a royalty attached.
I like that. I like that answer, Suzy. You snapped into businesswoman mode there.
I have to remember that I'm not just a scientist these days. I do have to keep an eye on the commercial angle as well.
Not quite right. It's not all about saving the world. You've got to save the world and make some money for shareholders. As I'm sure they're all listening to this, going, yes, absolutely. Right. Okay. Let's ask about TheoremRx and the licensing agreement. Now we've got questions from Andrew Selman, Alan Zeng, Nicholas Slater, James M. Who hasn't asked us questions about this? To be frank, they're all much of a muchness. They're all sort of. I think these two questions that we've pulled out pretty much encapsulate them all. What percentage chance would you give of the sub-licensing agreement being completed for 201, and why?
Well, I think everybody knows that I believe TheoremRx are the right partners to take ValiRx forwards. They have the right level of expertise. They have the expertise in the science and the commercial angle to find further partners, and in the financial angle, actually, to understand the money that needs to be raised to take it forwards and to do that. Now, it's disappointing that the timing hasn't worked in our favour on this. It would have been nice to have this wrapped up a year ago, to be quite frank. But they are still the right partners. They are still the right people to do it, and they are still moving forwards with it. So while they still have a credible plan in place to get that funding, I am completely backing them on it. So okay, I believe they will do it.
Which takes us to Alan Zeng's point. Have they done enough to justify exclusivity?
If they hadn't, we would have taken it away from them by now. If they weren't demonstrating credible progress to us, then we wouldn't just remove exclusivity, we would remove the letter of intent, because there's no point in having the letter of intent there and the intention to sublicense to them without the exclusivity, because they just wouldn't be able to do anything with that. So it is very much all or nothing. They have the letter of intent and they have the exclusivity until we believe that it's no longer relevant for them to be holding it.
And the sense that I'm getting from the questions that are coming in is that some of the shareholders would like to see a little bit more about what TheoremRx are actually doing, some of the work that they're doing. Is it possible to actually publish some of that, or give a little bit of clarity as to what they are doing? That's me channeling my inner shareholder there.
So very good job of channeling the spirit of the shareholders there. Now we do understand that everybody is frustrated with this. We understand that they would like to see everything that we see on it. But I think we've explained before that the work that we're doing with them is very much two-way. So the things that they're giving us scientific input, well, they're asking scientific questions about 201 that then allow us to take things forward on about 301. So it is a two-way process how we're working. They're working with our patent agent already, but they are not doing any physical science in the lab yet. So I know I've had questions about this over email before, where people have said, well, have they done this, that, or the other experiment yet? No, they're not doing anything like that. They are spending time and thought and helping in an intellectual manner, but at the moment they're a startup without funding. If they had funding, they would have signed the sublicense and paid us already. So there is no question about that whatsoever. We believe they will get that funding, and as soon as they do, we will have the sublicense with them. But okay, it's a belief. Until everything's signed, it's not signed.
Okay, Paul Newham again. He asks, can we expect an update in the coming weeks from the Hokkaido evaluation? You said that was due to end at the end of the year, December, did you?
Well, it's the middle of December that's scheduled to be on the 1 year point. So yes, we will provide an update before the 1 year point comes up.
Oh, does that mean you're not going to give us any hints and tips?
It's science. We're still waiting for data to come in. We're still working on it. We just have to see where we are at the 1 year point.
Okay. And work in progress.
Exactly.
Work in progress. They've got a great name, Hokkaido. Is that the university?
Oh yeah, so Hokkaido is an area of Japan that has a university. So they are a very strongly scientific university actually in Hokkaido. We've seen a number of projects from them.
Very good. Imran Khan asks, has your daily work changed since you've added 2 additional staff? Do you get more free time to do scientific work, for example?
Oh, right. The scientific work I do at the moment, unfortunately, isn't in a lab because we don't have a lab. But yes, it's been absolutely brilliant having the 2 new staff members. Stella, of course, has come in place of Kevin Alexander, the board member. So that's not so much. Although Stella has a very different focus to Kevin, so it's really great working with somebody with that much energy as well as that much experience. But working with Kathy has enabled me to actually have more headspace to think science. So instead of having to store all of the science in my own head, I can talk to Kathy about it. And it's absolutely brilliant because she understands how I think science, which isn't always a given, and she knows how to push back on my understanding of science. And she gets all of the commercial angles as well. So it's having Kathy there as a right-hand person for the last few months has been absolutely brilliant. And it has given us a lot more scope. It's a synergy thing. It's not just there's another one of me to do more work. We can actually build something greater together.
Oh, that's a very good answer. Finally, Andrew Selman again. Can you tell us if any big pharma companies have shown an interest in the KC-TNBC? I'm not sure I even understand what that is, but hey, you will.
Well, yes, no, he's referring to Cytolytix. So have we managed to...
Oh, Cytolytix. All right, I understand what that is.
Of having it. Well, so we've had Kathy and Mark Chahan have been out at ValiEurope last week, and that we did manage to announce the Cytolytix launch on the first day of the ValiEurope partnering events. That was brilliant that they were immediately able to go out there and start talking about it. So yes, actually we have already presented it to quite a few biotech and pharma companies. So that has been really good to have a new project to talk to everyone about. We haven't sold it yet, but we do intend to do some work on it, so to build some value before we sell it on.
Brilliant, Suzy. That was 30 minutes well spent, I have to say. You were fantastic. That was fantastic. I love the slides, and that was hugely engaging. That was terrific. I really enjoyed that. Thank you so much.
Thank you for the invitation, Donald.